Hey everyone, welcome back to Manin's Medical Academy. Let's make learning science easy for everyone. Today we'll talk about the second part of protein synthesis, which will focus on translation, or how genetic information in mRNA is translated to proteins. Let's get into it. After transcription and the 5' capping, the 3' poly A tail, and splicing, we have a fully mature piece of mRNA. As we talked about previously in my other video about transcription, which by the way, if you haven't seen, I recommend you check that one out too, since these two videos go hand in hand. I'll also link it in the description below. But as we talked about before, mRNA carries genetic information that is ready to be eventually translated into protein. So now, the mRNA leaves the nucleus to find a ribosome. Ribosomes are protein-making machines that are made up of rRNA and other protein subunits. The R in rRNA stands for ribosomal, and it is produced within the nucleus, which then forms the ribosome. So, in eukaryotes, once the mRNA leaves the nucleus and reaches the ribosome, whether that is a bound ribosome, which just means the ribosome is attached to the endoplasmic reticulum, or a free ribosome, which just means the ribosome is free-floating in the cytoplasm, the mRNA attaches itself between the 60S and the 40S ribosomal subunits, and now the real fun begins. As the mRNA fits between its ribosome, it is translated with a sequence of 3 nucleotides at a time. This is also referred to as a codon. mRNA is read by another molecule known as tRNA. The T stands for transfer, because it transfers amino acids to the ribosome. The way that tRNA knows what amino acid to bring to the ribosome is because it reads the mRNA sequence with its own 3 set of nucleotides known as an anticodon. In simple terms, the tRNA anticodon reads the mRNA codon and transfers the amino acid to the ribosome, and then the tRNA leaves. Then, the amino acid then folds up and produces a protein, which I'll show you guys in just a minute. There are 3 sites in the ribosome where the tRNA attaches: the E site, the P site, and the A site. The main thing you need to know is that the initial tRNA, the first tRNA that brings its amino acid, binds to the P site. But obviously we're not done yet. We need multiple amino acids to make proteins. So now, the next tRNA that will come along attaches to the A site and brings its own amino acid with it. Both of these amino acids will now form a bond with each other. This bond is also known as a peptide bond. And after this, the ribosome moves down along the mRNA molecule. The ribosome moves in a sliding format, so it makes the tRNA in the P site move to the E site and it'll exit the ribosome. The tRNA in the A site then slides over to the P site, opening up the A site for the next tRNA to bring its own amino acid to form another peptide bond. Then eventually, these tRNAs will move to the E and P site and the process goes on. Eventually, the ribosome will reach a stop codon, which tells the ribosome that no more amino acids will be added to the chain and that it can leave. After the ribosome leaves, the amino acids will fold up into a protein. And as it folds, the amino acid evolves through various primary, secondary, tertiary, and usually quaternary structures. The protein will then exit the cell through a vesicle secreted from the Golgi apparatus. The protein will then perform its own function, whether it's to provide support, repair muscle tissue, make hormones, etc. This process is responsible for functions like insulin production or muscle hypertrophy, so make sure you're getting enough protein in your diet and getting your gains. If you're new here and you want to learn more about biology, anatomy, and just all things science, start now by subscribing. And if you like this video, make sure to hit the like button, and we'll see you in the next one.